13 or 14 people on the line. I think we can probably get started. I don't think this is going to take the full hour, but we'll see. So as usual, the agenda, I'll go over a bit of what's gone on the project over the last couple of months. And then I'm going to turn it over to Jose, and he's going to update on the mass spec imaging in the brain tissue. From there, I'll turn it over to Ahmed, and he's going to give a bit of an update on some recent ADME and DMPK experiments that were run. And then I'll get into some of the IND-enabling activities that have happened, particularly starting up the CMC program and some discussions we had on clinical planning. Yeah, so since the last meeting, we have done a little bit of ADME and DMPK. What we did do was we ran a rat excretion study. So it's a cannulated bile duct study. And I had Mike and Divya and Liz from Trials review the data, and they commented that it only shows that a small portion of drug is unchanged and recovered in the urine and the bile. So this indicates that we're seeing metabolism rather than or just biliary or renal excretion of the product itself. So that's the predominant route of elimination. They did comment that the renal clearance is broadly in line with what they predicted based on glomerular filtration rate and free unbound M4K2009 in plasma. So that suggests that any renal metabolism is passive rather than active. We also saw a small amount in the feces, so this may be that we're just seeing some unabsorbed drug being excreted in the feces. We also ran a powder and capsule PK study with our crystalline dihydrobromide salt. So this is the first time we've actually dosed an animal with a solid dose of M4K2009. So we did see high bioavailability, which was nice with this salt form. We don't see as high exposure as we do with a liquid formulation. That's not surprising. But it's the good take home is that we do still see high bioavailability when we dose it as a solid. So for CMC, we've just really started the process development work with the CRO over the last 2 to 3 weeks. And really their initial focus is on optimizing those first 2 steps, which are both palladium catalyzed cross-coupling reactions. So they've been looking at a few different catalysts, solvent conditions, and temperature to kind of optimize those 2 steps. And finally, we had some I would say, very positive discussions with the Connect Consortium regarding clinical planning. We did discuss a little bit the potential of a phase 0 in adults study. But I think we came to the conclusion that that's probably not something that we need to pursue at this time, because they were able to arrange an informal meeting with us with the FDA as the Division of Oncology. And Max and I took part in that meeting, and I would say it was very positive overall. They were very supportive of what we're doing and certainly encouraged us to ask questions, as well as apply for a rare pediatric disease designation as soon as we can. So they are supportive of first-in-child clinical trial, provided we have suitable evidence to continue in that direction. And they've really encouraged us to continue to ask questions, specifically through a program that they have called an Interact meeting, which is essentially a pre-IND meeting, really specifically for groups like us that are really in the early stages of preclinical development. So with that, I'm going to turn it over to, I'm going to stop sharing and turn it over to Jose for him to present some work on his mass spec imaging.
Okay, yeah, thank you. So it's going to be very fast, but I think that I wanted to show you what we've done because there are some problems here we have to solve. It's a pity I didn't see Rebecca among the people that is
Yeah,
assisting to this.
unfortunately, she couldn't make it today.
It's a pity because, well, we received three samples, one treated with M4K 2009 and two other samples from animals treated with the vehicle. And the first problem we found was that the samples were really deformed. I mean, you can hardly recognize a brain here. This one, that is the treated one, is just half of the brain. Well, you can recognize the brain here, but still, it's hard to get good sections out of here to map the compound here. So we, despite these problems, we got the 16 micron thick sections to we placed them in ITO covered slides, and we set up the conditions to try to detect the drug and to to do image. So we run three sets of experiments. First, we try to directly detect M4K 2009. Then we try recrystallization to increase the signal intensity. And then we did some spotting to see the limit of detection directly on tissue, because it's different from just the limit of detection of the drug just on a multi-target. So this is the first try. You can see a beautiful spectrum of the lipids in the tissue, but the M4K is supposed to be here. Well, you can see this is the matrix. It's a very small peak, so this certifies that the sample protocol was correct. M4K 2009 should be here. When you zoom in, you have to zoom in again, and it's not this peak here, it's this peak here. We got 90,000 absolute arbitrary units of intensity, and this peak is about 2 units. So it's hardly above the chemical noise that is this here. When you try to do the image, well, it's just randomly distributed across the tissue. Of course, in those spots, probably there is an increasing concentration, increasing signal. Now here, because this is the average over the whole tissue. But I think that we are just in the limit of detecting it, and this is 100 micron per pixel. So this is, it's not a good, it's a bad spatial resolution, bad for our standards. If you want to compare, this is cholesterol that Rebecca asked us to have a look at. You cannot recognize the structure, the histology of tissue, but cholesterol gives slightly less than an 8% signal compared to the strongest peak. So it's detectable and we can see it, and image its distribution along the tissue. So when we do we know that this is 2009 M4K 2009? Because in the vehicle treated tissues it's not present. This is the as I said, the chemical noise probably limit. And well, this is the distribution of cholesterol in the vehicle treated samples. In this one, you can see some structure. This is the corpus callosum. This is the white matter in the hippocampus. In the cerebellum. But in this one, you cannot recognize the histology. It's just too deformed. So we recrystallize. It's a very simple procedure. You just get a Petri dish, you attach your sample upside down to the lid, and you put some acetonitrile. Well, in the original receipt, it says to heat up to 50 degrees, but I think that's too harsh. So we just leave it there for 10 minutes at room temperature, and we close the observation window to just to 350 to 550 to avoid saturation due to the strong peaks from the lipids. And there we go. This is where M4K 2009 should appear. That's the peak. So we had an increase in signal intensity. Here we have now 11 arbitrary units of signal intensity, which is good. Those are the small peaks that were present, I mean, the chemical noise that is present in the sample. But distribution, well, you cannot, I mean, this is just kind of randomly distributed across the tissue. Now, if you do the image of cholesterol, you can see that it's delocalized. So the recrystallization procedure structured the sample that's extracted the M4K 2009 from the tissue, helped us to make M4K 2009 migrate into the matrix, increasing the signal intensity, but at the same time it produced delocalization of the lipids. So then we started spotting it on the tissue sections to see the limit of detection. And well, these are the spots, and you can see that there is more M4K 2009 outside the tissue. I mean, the signal is stronger. It's not that there is more. The signal is stronger because, and this is probably telling us that there is ion suppression effect inside the tissue because the lipids. So having lipids there, I mean, just putting the M4K 2009 on the tissue, on top of the tissue, is decreasing the limit, I mean, increasing the limit of detection. So we try to do a quantification. In another two sections, and you can see that, well, we put, I mean, 8 times 10 to the minus 6, minus 5, minus 4, and you can see, you can guess which spot corresponds to each of the concentrations. Still, as I said, MOLDI is not a good quantitative technique because the signal intensity will depend not only on what you spotted, but also in the tissue that is underneath, right? And actually, well, there is a kind of, I mean, I just draw these lines, okay? There's a kind of I would say, linear behavior of the signal with the concentration, but there is a large variation of the signal intensity depending even on the sample that were spotted. If you look at the lower concentration we spotted, actually, if you play with the scales and you saturate the scales, actually we can see, I mean, we have no problem detecting this amount here. Those are the spots with the smallest amounts, and you can, yeah, detect it. Here is that. Of course, this is too strong. It's 2 orders of magnitude stronger than this one, so that's why you can hardly see this spot. But once you play with the scale, you can see it easily. Okay, so what is my summary and what I think the next step should be? So we can detect M4K 2009, but with very low signal intensity. Now, how much time passed since drug administration and animal sacrifice? There was this, this piece of paper with the samples, and I don't know if this is true, but it says death date December the 2nd, 2025, and it was 22 days post treatment. So I don't know if actually what they sent us were animals that were treated 22 days before sacrifice. If that's the case, this is a tremendous success. But if these animals were just treated and they were sacrificed, this is a terrible problem. So that's one of the questions I have. I mean, how much time did pass since injection or drug administration until sacrifice? Well, we detect cholesterol. That was one of Rebecca's questions. We can detect it and we can image it. And we can go below beyond the spatial resolution I saw you, because we are not using the whole pixel for recording the images. Recrystallization increases the signal intensity, but also it causes lipid delocalization. No, yeah. So maybe we have problems making M4K 2009 migrate to the matrix to detect it. I don't know, but the protocol seriously compromised the image quality. We need to improve sample handling to avoid tissue deformation if we want to see the histology of the tissue. And well, we're looking at 2009, but remember that 2281 had a better limit of detection, so that give us good chances to detect M4K 2281. That's it.
Right. I have a few questions, Jose. Thank you for presenting. Yeah, it's very nice. One problem, I think the brain you showed us at the beginning were quite deformed, and that's we normally when you do a nice extraction, it should not be like that. It should be you should have a structure, and you should cut it, and you should. But anyway, that's something people who collected them probably not here today. So we can discuss that when with them when they're here. But for also the other important factor you pointed out already, we asked for dose and within 2 hour collection post dose brain. So that was the hypothesis, and that was the protocol we requested. And if it is, as you said, it's not clear from the sample description you have in hand, so that's also a problem.
Okay, let me stop sharing. Okay, what we got is this.
Sorry. Okay. Okay. Yeah.
From here is I try to understand from here. I mean that there is pedigree, ear tag, treat cage, treatment, sex, death date, days post-treat 22, days post-injection 49, weight loss, yes, neural symptoms, no, tumor unrelated, no, comments, weight loss, blah, blah, blah. Experiment start date, that was in October. Dosing starting date, that was in November. Last dose was, okay, last dose was December the 2nd. Oh, my birthday. That
And the
was
sample
okay.
collection
Yeah,
was
yeah. Okay, last dose was December the 2nd. Death date was December the 2nd.
okay.
So maybe, yeah,
Is character
they were
affected? Yeah.
sacrificed the last day,
Okay.
a couple of hours after those. So that's bad, because that means that they were administered and killed the same day. I expected it to be killed uh, a few days after dosage.
And also my last thing I was going to ask you, when you are doing this section, do you, I mean, I'm not sure if you have a pathologist, or can you see where the tumor is or no?
We didn't do any staining, and according to Rebecca, the tumor was it doesn't make a solid tumor.
Okay.
It just spread along the whole tissue. We didn't do any staining or anything.
I'll stop here.
Okay, thank you.
Yeah, I just had one question. I seem to recall when you were doing the method development with 2009, you saw the parent, but you also saw some fragmentation. So is that included in your analysis on this one,
Yeah,
on
we
this
looked
set of
for
experiments?
yeah, we looked for everything. Yeah,
Okay.
but we didn't see anything else. I mean, we were just scanning the three spectra from T1, V1, V2. We were moving all the spectra and looking for new peaks in T1. Nothing really remarkable.
Mm-hmm.
Yeah, I told you, Ahmed, that this is a very difficult experiment.
I know.
Yeah, I don't know what else to say.
Okay. Yeah, I mean, it would be nice if we can do some n equals 2. And so far, what we can say right now from the very preliminary data you have just from one brain, that it is not localized in one spot. Like whatever you can see, it's spread out over the whole brain. That's probably what you can say right now,
Yeah,
just from this one. So, but it would be nice to have a intact brain where you have more. You can see the shapes and all this distribution. And then if you do a slice and look at this distribution again, that would be ideal. I mean, you already nailed all the experimental procedure, like detection. I think the recrystallization may be a way to go. I mean, if you don't care about the lipid localization, then recrystallization, when you are seeing the 11 arbitrary unit or whatever, that would be probably the way to go. I think that is what we are saying is probably the number we are getting in the brain. Like that's the maximum number. I think if they did uh, dosing and collection after 2 hours, that's probably the Cmax we see in the brain. So if we can see that, and then if we can see some sort of localization in a brain where it is not mushed up like the one you have, so maybe that will be better. So, but this is something we can discuss offline and see how can we generate that kind of brain.
Mm-hmm.
yeah. Also, I'd love to look at the other compound because we have a lower limit of detection for the other one.
Okay.
Yeah, I guess, well, this is a qualitative, but it certainly doesn't seem to match up with what other data that we have showing that we do see exposure in the brain. Whereas this with this, I guess the difference is we haven't really looked at it in treated animals, which in this case it is. A lot of that data comes from healthy animals, but
Yeah,
certainly
but
we do.
yeah, I mean, what I think, Pete, I think we are seeing the compound
Hmm.
in the brain. But the way, like, if we are using MALDI, a different type of detection. One thing what we can do, we can like after Jose do the sectioning, we can take that other brain and do a homogenization and do an LCMS, regular LCMS, and
Ah, yeah,
figure
yeah.
out what is the concentration we actually have in the brain, right? So that's something, I mean, we can do this fine detail later, so we can figure out and have another protocol. And once we, or maybe we can have three mice, one mice we can give to say, and two other we can just do the quantification in our hand by LCMS and make sure that we have adequate concentration there, right? So those kind of
Yeah.
analysis we can have, and then we'll be sure that this is the concentration limit what Jose is seeing by his
Yeah,
method.
yeah.
Yeah.
As
No, I think
as I
that's a
said,
really good idea.
yeah, it's a good idea, very good idea. But as I said, the problem is that what you're detecting is what is migrating to the matrix.
Hmm.
So yeah, when you spot it on top, you deposit the matrix on top of the tissue, then yeah, it's easy to detect, despite the interference with the lipids that are already in the tissue. But the problem is that probably the drug is inside the cells. And so it probably is, or maybe is docked into a protein or a receptor. I don't know. I mean, you guys know where it should be. So probably is difficult for it to migrate to the matrix. I don't know if they're naturalizing the tissue, for example. The naturalizing the proteins would help the drug to migrate to the matrix so we can detect it. I don't know. I don't know
Yeah.
if you come out with some different strategies.
Yeah, I think we can discuss more and figure out if there is any other strategy we can apply to see more quantity. But whatever you can see for now, what we can say safely that it is distributed, at least in the tissue you saw, it's distributed almost evenly in the brain, right?
Yeah.
No specific region. That's what we are seeing.
Yeah, I would say so. Yeah, we can scan, as I said, we can go to 50 micron, and probably have the same signal intensity, because the laser on this equipment is designed to have a good spatial resolution. So it doesn't really use the whole pixel when you say 100 micron. This is something we discovered just doing these experiments. We can probably go to 50 micron with the same signal intensity. But as these were just some tests, we didn't run that test. But we will next time we will go to 50 micron or 25, see if there is when we have a signal intensity decrease. So what I mean is that data could be more informative.
Okay. Thank you very much, Jose, for all your work. I'm going to move on to.
Can you see my slides?
I'm going to. Oh,
yeah. Oh, you're sharing it?
Yeah.
Okay, great. Okay, so we recently did plasma protein binding, dog plasma protein binding of 2000 M4K 2009. What we can see that this is a very high free fraction. Normally, this free fraction, like we were seeing for mouse and human is in a 5 to 9%. But here we saw about 20% free fraction, which is significantly higher than human and mouse. Actually, I was looking for the rat data, but it looks like maybe we don't have the rat data for the plasma protein binding. That's something we need to have, because both dog and rat are going to be our tox species in the future. So this free fraction may have an impact for that. So this is just to keep in mind in the future. Okay. Okay, and then next slide is Pete. I will walk through it slowly. Pete just mentioned that we had powder in capsule. This is for the first time we dose powder in capsule in mice. And we did, and this is the 2 HBr salt that we used for this experiment. The dose for IV was 1 milligram per kilogram, and PO was approximately like there's 3 mice. Both 2 mice got 42, and the third mice got 43 milligram per kilogram. This is based on the weight of the capsule, and the half-life was pretty good. It's consistent with what we have seen before for the PO. The IV half-life is also very consistent with what we have seen before in our other experiments. We have the AUC, which is respectable from this dosing, and about 90% bioavailability is what we saw from this powder, which is very good compared to dose when you are dosing powder instead of a solution here. So if you want to see, if you go to the next slide, if you want to see where it falls, if you are comparing with these liquid solution dosing, this is the solid dose, slightly below the linear range. So it's slightly lower than Like if you had the same dose in solution, it probably will be somewhere here. But from powder, you have to think about the other distribution, your solution and distribution. So this is probably not bad compared to the liquid what we are getting, like solution formulation. So that's pretty much, and the data is all presented here in this slide. You can have a look. Do I have another slide? Can you go to the next slide? Well, that's just the data from the other, just the solution one. Yeah. So if you can go back, that's it then. So that's the data we got from the powder in capsule.
Does anyone have any questions on those experiments? Okay. Well, if not, I'll move along to some of the IND-enabling work. So I mentioned we're running this scale up at a CRO, and currently they've been working on it now for just about 2 or 3 weeks, really focusing in on these first 2 steps, which is a palladium catalyzed cross coupling. They've been looking at different catalysts, different solvent conditions, because one of the issues with this is you will get dialkylation. So basically this aryl group adding to this, also to the second position on this pyridyl ring. So they have gotten some conditions, and now they've scaled that up into multi-gram scale. And so far, I'd say it's looking quite good. And then they're also moving along to this second step, again, looking at different catalysts and solvent conditions to optimize this reaction before they proceed to scale up. With this last step, which is removing this protecting group, the procedure that we have right now is we do this in 2 steps. We remove this with HCl and then make the free base, and then make the HBr salt. What they're going to look at is whether or not you can just go straight to the di-HBr salt in 1 step. So that's something that they'll be starting probably in the next week or so. They haven't really done any work on analytical method development at this point. They're really waiting till they have some material in hand to be able to do that. Any questions on the scale up?
Are they going to look at the palladium content?
Yeah, so we sent them some samples that we made in-house. And I just followed up with them yesterday to see if they had run, to see what we're dealing with the basically the lab scale. So I don't have those results yet, but yeah, that is something that they will be looking at, but it's not any data that I have currently. Hopefully by the next update, we'll be talking to them on Thursday, and I'll see when they can generate that data. But yeah, it's definitely something we need to be cognizant of.
Thanks.
Okay. Yeah, so the last item is really clinical planning. So I mentioned at the top, we've had some discussions with the Connect group on what what a potential trial would look like, and we did consider a phase 0 trial in adults. That would just be the first time we would dose 2009 in healthy individuals in order to generate some human PK and potentially extrapolate that to a first in human dose. We discussed that with the group at CAMH, whether or not that would be something that they could do, as part of their work on tracer development. But with those discussions, we decided that a particular phase 0 with M4K2009 is different from what they would be doing as a tracer would really be looking at plasma PK. So it's not something that they could do, but if it, if we did need to go down this route, there are other groups that we could approach for a phase 0. But after those discussions with Connect, we basically kind of decided that that would be, a second, a fallback position. But right now we feel that we can move forward with hopefully move forward with a first in child trial. So the Connect Consortium was able to get us in contact with this group at the FDA. We had some discussions with them, and they certainly were supportive of a first in child clinical study, potentially starting with older children, say 12 and up, as the first cohort, getting human safety and PK data in that group, and then moving quickly into younger children once a suitable dose is established. But one other, I think, interesting takeaway from that meeting was they don't require us to do our tox study in juvenile animals. They said we can get data in adult animals. So I think, that's certainly going to make things a little bit more straightforward. We can do a typical toxicology study and not have to worry about the timing of juvenile animals. So I thought that was a really good feedback we got from that meeting. And I mentioned, they really encourage us to take advantage of their Interact program for follow-up discussions. So I am currently putting together a briefing package to submit to hopefully put in place one of these meetings. If anyone on the line has experience with briefing packages, FDA meeting interactions, please reach out to me, after the meeting. I really appreciate some of your input on what I'm putting together. We'll be putting this in with Connect as well. Obviously, they'll be taking care of the clinical section of the briefing document, but certainly any input we have from our internal team would be greatly appreciated.
Peter, it's Alex. What children over 12 would you treat? I mean, most of the DIPG patients are younger.
Yes, so we did have those discussions, and I think it would be, so they wouldn't necessarily need to be ACVR1 mutants. I've also had some discussions with Matt Dunn's group in Australia, and he's a big proponent that BMP signaling still plays a significant role in DIPG, whether it's ACVR1 mutant or not.
Yeah.
So I think the idea would be these would be relapsed patients that we could use in that cohort. Again, not necessarily ACVR1 mutant, simply to get that safety data and first in human before we moved into a younger population. And the main reason being is the risk of giving a subtherapeutic dose to a young child. So again, these are all kind of ideas that we're throwing around for that trial. But any other additional input on how we design that trial, I think there's still some questions, and what that cohort eventually looks like is still a big question. David, do you have a question?
Yeah, Pete, you may have said it, but so what's the main criteria for determining a quote unquote suitable dose?
That's a good question. I think it's more on the the safety aspect, and then in conjunction with what a predicted efficacious dose would be.
Based on plasma concentrations.
Exactly, yeah.
Okay, so we would be inferring what the brain concentration is based on plasma concentrations from various
Yeah, so
preclinical
I think we
experiments.
have some predictions that Liz has done on what an efficacious dose would be based on the animal data that we have. I think we'll certainly strengthen that data once we look at exposure in our tox study. But I think those are kind of what you would be looking at, once you start planning for that trial. But again, these are all things that we'll need to work out with the Connect team.
Peter, I
can,
think I was just going to say, I think the idea was dose escalation cohort at the beginning of the trial. So there'd be some dose finding element to it, even though it's, in a patient cohort.
and
Sorry if that
would
was
we
already
be able
clear.
to? Oh no, I didn't. Yeah, thank you. Actually, that's helpful. I'm just curious also about allometric scaling and what would we be at the start of the trial in a position to make accurate predictions about what we think is going to be the dose that delivers this plasma concentration that we think is needed? I guess maybe that's a question for Ahmed.
Well, yes, we have to do allometric scaling, as you said, David. I mean that will give you at least the range, and as Max was saying, this is why actually in the clinic we want to do a multiple dose, making sure. And you also said an important point here, like we are going to base everything from the plasma concentration. If you want to do it from the brain concentration, it will be very, very, very difficult. Like even from animal to human, like human, and it is going to be very complicated to do that. So it will be best to do it based on the plasma concentration, as you mentioned, and for many other reasons. And then we are going to do an allometric scaling, obviously, to predict the most probable dose that should be going in the human. And then tox is going to guide us, right? What will be our starting dose? What will be our lowest starting dose? And then how far we can go from there, right? So this is all has to be done.
Thanks, Ahmed. And I guess part of my question was, do we have all the data we need to conduct the allometric scaling? Are there key studies that we need to do preclinically to get that information so that we can do the allometric scaling?
So more species you have, better prediction you can do, right? So if you have like only one or two, you only have 2 data points. If you have more species in between, you have more data points. So that's
sure. So what species do we need, or what's the plan to get that data?
So like you need rodent, non-rodent, and then you can go for human, obviously. So, but if you want to add NHP, and I'm not sure if that's the plan, then you will have better prediction from the NHP also, right?
I don't think we had considered NHP.
Sorry, Pete, you say we have not?
We haven't considered a non-human primate PK.
So we would just need rat and dog.
Rat,
2
dog,
species.
and mouse. Yeah, and
mouse.
Yeah.
And of those, we still need to do the dog.
I think dog is done.
We have dog PK.
We have done dog. Yeah.
Yeah. So essentially, the answer is we have all the data that we need to do the allometric scaling. We could do it today if we wanted to.
Yeah. At least the preliminary prediction we can do. Yeah.
Okay.
I think Liz has been looking at human dose predictions and kind of doing a bit of simulating herself as well. So I'm sure any other further information she can maybe go through one of the next meetings when she's back.
Thanks,
Yeah,
David. That'd be very interesting.
Yeah. Okay.
Max, did I miss anything from the discussions we had with the FDA and Connect?
No, I think that was a great overview. Just a matter of getting back in to have that interact meeting and get some more specific advice.
Yeah, so we're putting together some specific questions, around mostly I think around talks and potentially the clinical planning. So, we can certainly share those questions. And David, if you have other specific questions that we could add into that meeting, we can consider them. Generally, you don't want to go in with a boatload of questions. I think they generally say no more than 10. So we kind of have to make sure that at least for this initial meeting, we're asking the most important questions, but they certainly would open it up. Like this isn't the only meeting that we could potentially have. They are, open to, further dialogue if we have other questions along the way, which, I thought was, was really positive that they actually encourage you to reach out to them.
I mean, I know the team may be budget limited, et cetera, but have you considered hiring a clinical pharmacologist consultant, somebody who could actually help you formulate these questions? Because it sounds like most of our expertise is in the preclinical space, and we would actually need to have that person on board.
Yeah, so I did put that in kind of the overall budget to hire someone in for certainly a group for toxicology consulting and regulatory consulting. We have spoken to Intrinsic in Mississauga, which is a group I think that OICR has dealt with in the past. So I have some kind of high level quotes on what they would charge for that. I think speaking with, we might also be able to get some feedback from the Connect team as well. But absolutely, you're correct. We're strong on the discovery side and early stage development, but the talks and regulatory, we definitely need to bring in extra expertise to help with that.
Yeah, it might be worth. We've interacted with somebody named Sam Blackman from Day One Pharmaceuticals. Of course, that's their space, and might be worth reaching out to somebody like that just to see if they have any thoughts or recommendations, and might even be able to work with us, because I think there's people out there who would be able to actually know exactly what to do in this situation. But like you say, going in to these FDA meetings, you want to have all your ducks in a row. You want to be able to get the most out of the meeting. You don't want to waste the opportunity to meet with them. So I think it might be important to really get that expert input before going in, as opposed to trying to rely on the folks here.
Okay, that's a good point.
Just to add to that, so the Connect Consortium, they're committed to being involved in these meetings as well. They have some in-house expertise in this that they don't want to sort of ask them to dedicate their full resources to this at this stage, but that they would sort of set up a meeting and potentially we can get some help there. I mean, the key thing is we're resource constrained, so we can't really spend $100,000 on a consultant just for the first Interact meeting. So we're kind of trying to limit the use of that budget as much as possible to maximize its how far it goes. That makes sense?
Yeah, that makes sense. Thanks.
We're just trying to scrap together as much as we can without getting rid of our budget, but where it's necessary, I think you're right. We have to make sure we're doing the right thing.
Thanks.
Okay. Well, I appreciate all the feedback. Is there any other comments or questions that people have? That was basically all I had to present today. If not, thanks everyone, and probably schedule another team meeting for, I would say, 2 months from now. I don't think we will have a lot to discuss on a month-to-month basis moving forward.
Great.
Okay.
Thank you.
Thanks everyone.
Thanks.
Thanks.
Bye.
Bye.
Thank you.